Gene therapies that can literally fix faulty DNA, immunotherapies that can supercharge our own cells to fight cancer, stem cells that can regenerate entire tissues. This stuff isn't science fiction anymore. It's the cutting edge of medicine. But every single one of these incredible breakthroughs shares one massive, yet microscopic roadblock.
See, it's not about getting a drug into your bloodstream or even to the right organ. The real challenge is so much smaller, but infinitely more difficult. It's all about getting past the front door of a single cell.
Because that's really what a cell is, right? It's this tiny little fortress, a high-security vault that's had billions of years to perfect its defenses. So to make these amazing new therapies actually work, we first have to figure out how to pull off the perfect biological heist. And our main target? It's the cell membrane. Now, this isn't just some simple wall. It's an intelligent, dynamic barrier. Its whole job is to maintain a very specific, very delicate world on the inside, completely separate from all the chaos on the outside.
So before we even think about planning our break-in, let's take a closer look at what makes this fortress so incredibly tough to crack.
Okay, so the membrane's #1 job is maintaining this wild chemical imbalance. Think about it. Inside the cell, you've got a ton of potassium and ATP. That's the cell's fuel. But right outside, it's the complete opposite, loaded with sodium and calcium. This precise imbalance isn't just a nice-to-have. It's pretty much the definition of being alive. If you disrupt it, even for a second, you risk killing the cell. The stakes here are just incredibly high.
All right, so we know our target. Now, what's the valuable package we're trying to sneak inside? Well, it's not just one thing. It's a whole arsenal of game-changing therapeutic tools.
And the range of stuff we're trying to deliver is just stunning. I mean, look at the scale here. Over on the left, you have these tiny drug molecules just a few nanometers wide. But as you move across to the right, we get to the heavy machinery, these massive protein complexes like CRISPR-Cas9, which are basically designed to perform surgery on our DNA. We need a delivery method that can handle all of it.
So what's the mission? What are we trying to do? We're breaking in to deliver things like cryoprotectants, sugars that can protect cells during freezing, which is vital for organ transplants. We're inserting proteins and antibodies to fight diseases from the inside out. And maybe most excitingly, we're delivering tools like CRISPR to directly edit the source code of genetic diseases.
So we've got our target and we've got our package. Now for the really fun part, the break-in. Over the years, scientists have come up with some incredibly clever and sometimes surprisingly aggressive ways to get the job done.
The first approach is exactly what it sounds like. If you can't pick the lock, you just kick down the door. These methods are all about physically tearing or squeezing a temporary hole in that cell membrane.
And the toolkit for this is just wild. Microinjection is the most direct. You're literally stabbing the cell with a microscopic glass needle. Then there's something called biolistics, where you coat tiny gold particles with your cargo and then fire them out of a gene gun. Seriously. You could even use cell squeezing, where you force cells through channels so narrow they get squeezed and their membranes just temporarily burst open. It's mechanical, it's direct, and you know what? It works surprisingly well.
Now, if brute force isn't really your style, how about something a little more elegant? This next method uses a powerful, invisible force to open the gates without ever actually touching them. We're talking about electricity.
This is called electroporation, and it's kind of like a controlled lightning strike on a cellular scale. By hitting the cell with a very specific electric field for just the right amount of time, and we're talking microseconds here, you can cause these temporary water-loving pores to form in the cell membrane. You can even tune the pulse to either target just the outer membrane or, with a stronger, shorter jolt, go even deeper and hit the organelles inside.
So the process is simple, but it's incredibly powerful. You apply the pulse, the pores open up, your cargo rushes in, and then, and this is the critical part, the cell's own repair systems kick in and seal the holes right behind it. It's a perfect temporary breach. Our third method is the most subtle of them all. This isn't about forcing your way in, it's about having a key. This is the biochemical approach, using molecules that are literally designed to talk to the membrane and open it up from the outside. This is just incredible. Scientists have basically repurposed toxins made by bacteria. These proteins are like little self-assembling nanobots. First, they just float around as these individual sleepy units. But when they find a receptor on the cell's surface, they latch on, they call over their friends, and they assemble into a ring. Then, all at once, they punch down through the membrane, forming a perfect, stable pore that lets our cargo flow right in. It's a beautiful piece of molecular engineering. But you have to remember, the break-in is only half the mission. A successful heist requires a clean getaway.
The cell has to survive the whole procedure, or the therapy is useless.
So how does a cell even know it's been breached? Well, the answer is calcium. Remember that huge difference in calcium concentration we saw earlier? When the membrane gets punctured, calcium floods in from the outside. That sudden rush is the cell's emergency alarm. It instantly triggers this complex cascade of repair machinery to patch the hole and fast.
So really, there are only two possible outcomes. In a clean getaway, the membrane reseals in seconds to minutes. The cell pumps out all that extra sodium and calcium, it gets its internal balance back, and it just gets back to business. But in a botched job, the damage is just too much. The cell can't reseal, it bleeds out its essential molecules, and it basically triggers self-destruct. It is a razor-thin line between success and failure. So after all of that, the high-stakes target, the mission-critical cargo, the daring break-in, and the clean getaway? What's the payoff? Well, the payoff is a complete revolution in how we treat disease.
We can now take an adult skin cell, use electroporation to deliver a few key proteins, and reprogram it all the way back into a pluripotent stem cell, which means it can become any cell in the body. We are delivering CRISPR machinery into a patient's own cells to fix the genetic mutations that cause diseases like sickle cell anemia. And in one of the biggest breakthroughs in cancer treatment, we're taking a patient's immune cells, delivering mRNA to turn them into supercharged cancer-killing CAR T cells, and putting them back in to hunt down tumors. This is not science fiction. This is happening right now.
Mastering the art of the cellular break-in has unlocked a whole new frontier in medicine. It lets us go way beyond just treating symptoms and start rewriting disease at its most fundamental level. So the only real question left is, what fortress will we crack next?